recorded, so everybody will be muted. If you have any questions during the webinar, please just type them in the chat box and we'll have time to answer them at the end. So my name is Malti Rahman and I'll be presenting today's talk, and I'm a product manager for Takara's cloning and protein expression and purification products at Takara Bio Europe. So Takara covers the Clontech, Takara and Salatus brands. Okay, so I'm sure you're all familiar with the clone, express and purify workflow for your protein expression and purification studies. So the first step usually involves cloning the gene of interest or your favorite target protein that you want to study further into the protein expression vector of choice, into your protein expression vector of choice. So most researchers use traditional ligation-based cloning for this first step, whereas today I want to introduce you to our next generation Infusion HD cloning technology, which is a really nice, innovative cloning system that is completely sequence independent. So once you've generated your protein expression construct containing your favorite gene of interest, you then want to express this target protein, usually with a tag such as a his tag, so that you can then purify it downstream. So to express this target protein, Takara actually have a wide choice of different systems, so not just E. coli systems, but also we have baculoviral systems, we have bacillus secretory systems, we have human cell-free systems, and I'll briefly mention these during my talk today. So once you've expressed your target protein, normally with a his tag, the next step is then to purify this target recombinant protein using the his tag, and we have many his tag purification products. So when you initially want to screen your clones for the best expressor of your protein, and to test the purification of different clones, you can use our next generation Capturome technology, which is based on high capacity membranes, and it's really great for fast screening. So once you've picked your best clone, you may then want to do a larger scale purification, and for this you can use our His60 nickel resin or Talon resin if you want very pure protein. So we really offer a nice range of products for the whole workflow, which should ease your protein expression and purification workflow considerably compared to traditional methods that most researchers use today. And this should enable you to reach your goal faster, your research goals faster, and publish faster. Okay. So first, before you start the cloning step, you first need to choose the expression system or vector that you want to use for your experiment. So at Takara we offer many different systems, including bacterial systems. So we have our very basic E. coli PET express and purify system, our cold shock expression system. We have a very specialized single protein production system which just expresses your target protein. Then we also have larger scale secretory system, so this includes Brevibacillus and B. satilis. Excuse me. And finally, we also have insect systems, so our baculoviral backpack system, and various mammalian expression vectors too. I won't go into these two systems into much detail today because it goes beyond the scope of this talk, but I will talk in more detail about the Brevibacillus and B. satilis systems from Takara. And finally, we also offer an in vitro system, so the human cell-free system from Takara, which is an in vitro transcription and translation system. So as you can see, the number of post-translation modifications does increase as you go from bacterial to mammalian systems. Okay, so now how do you go about choosing which system is the best for your specific needs, for your specific experiment? So this table on this slide should help you to choose the best system for your experiment. So if you're new to protein expressions and purification studies, Our first choice would be the E. coli PET Express and Purify system because it's a very simple system to use, and it's based on the IPTG induction of your target protein, and you get a very high yield, so up to 1 grams per litre. Number of post-translational modifications are few with the system, and it's also not secreted, and it's also, it's not good for temperature-sensitive proteins, but if you just have a very regular protein, this would be the first system to try. Now, if this doesn't work, then you could consider using our P-cold vectors. So this is also a bacterial system, but this uses the cold shock promoter. So you get expression or induction of your target protein expression at 15 degrees C. So although the yield is lower than the PET Express and Purify system, it's good for temperature-sensitive proteins, and also because of the lower temperature induction, the host protease activity is reduced. So if you're having problems with the PET Express and Purify, you could try our cold shock vectors. And again, post-translation modifications are few, and this system doesn't allow secreted, or it doesn't secrete the proteins into the medium, so you would have to lyse the cells. And then we offer a very special system called a single protein production system. So if you're really having issues expressing your protein, then you could use this. So it's a bit more complicated because here, basically, you need to change all the ACA sequences within your target gene before you use our system, which then basically cleaves all the mRNAs within the cell that contain ACA sequences apart from your target gene. So initially it's a lot of work, but once you have it set up, you basically just express your protein. So 90% of the total protein will be your specific single protein. So if you have a particularly tricky protein, this could be of interest to you. I'm not going to talk about this in my talk further today, but you can find out more information on our website. And with this system, the yield is low, but you get a very pure protein. Post-translation modifications, against they're few. It's not secreted, and it's also not temperature sensitive. Now, if E. coli isn't working for you, so for example, if your protein has disulfide bonds, then you could consider using our Brevibacillus or Bacillus systems, because they're also bacterial systems, but because it's bacillus, you get disulfide bond formation. And both are simple systems, and give high yield, so up to 4 grams per litre or 25 grams per litre. And as I said, post-translation modifications such as disulfide bonds are possible, and both allow secretion of the protein, so you get your active protein being secreted. You don't need to spend a lot of time lysing the cells. And with the Bacillus system, you can actually optimize the secretion. So with Brevibacillus, you can also express temperature-sensitive proteins because the proteins are expressed at 30 degrees C. Whereas with Bacillus, this isn't possible because the temperature is always 37. And then we also have a baculoviral system. So here, if you're more worried about post-translational modifications, this could be a better choice than a bacterial system. And you still get quite a high yield, so 0.5 grams per litre. And again, the protein is secreted into the medium, so purification is also easier. And it's temperature sensitive. And finally, we also offer our human cell-free in vitro protein, in vitro transcription and translation system, which is also a system. So although the yield is low at 50 mg per mil, and although post-translational modifications should be possible, but we have not tested this in-house, it's good, for example, for toxic proteins. So if you have a very specific protein and you don't need a high yield, then this is a good system. And it's also good for temperature-sensitive proteins. So the two protein expression systems that I'll talk about further today are the two bacillus systems, because this is something quite unique that we offer in the market. Okay, so what's the first step? So you've first decided which expression system vector you want to clone into. The next step is then to generate your protein expression construct. And as I said, normally most researchers would do this using traditional ligation-based cloning. but this, although if it works well, you can get your clone within 2 days. Often it doesn't work well for all constructs, and sometimes it can take months to obtain your construct. And you're often limited by available restriction sites, whereas with infusion this is never an issue. And you often also need to screen many colonies before you obtain your positive clones. And again, this isn't an issue with our infusion system. So now I'm going to talk about our next generation infusion cloning technology, which can really speed up your construct generation. Okay, so as I said, in most molecular biology experiments today, including protein expression and purification, the first step is a cloning of your expression construct. And you don't want to spend so much time on this step because it's not the major focus of your experiment. Rather, you want to spend as little time as possible on this step and then move on to the more interesting functional work. So imagine that you have a system that allows you to clone both single inserts and multiple inserts in a single step, that allows you to do mutagenesis, including substitutions and deletions, as well as clone small C-terminal and N-terminal tags, really all in a single step. A cloning system without any limitations. Well, we have such a system with infusion. We really believe that infusion is a new way, is a different and a better way to clone. So what's so special about infusion is it's sequence independent, so you can really use it to clone any insert into any vector at any locus. And this cloning is also seamless, so you don't add any extra bases to your construct, so it's scarless, unlike, for example, gateway recombination cloning, where you always get the recombination sites being added to your expression construct, which may result in additional amino acids that you don't want. The cloning is also directional, and you can also do many different applications with infusion, such as mutagenesis and also multiple fragment cloning in a single step. So cloning is always a one-step cloning process. So the beauty of infusion lies in the fact that it's sequence independent. So you're not restricted by the sequence of the vector or the sequence of the insert. So how does this work exactly? So the first step involves in choosing where you want to clone your target gene. So once you've chosen the locus, you then linearize your vector. So you can do this either through restriction digestion if you have sites which are available at the locus that you want to clone, or you can be completely restriction site independent and linearize your vector by inverse PCR. And if you use one of our high-fidelity enzymes, this is no problem at all. You can really be restriction site independent. You just need to know the 15 bases that are found at either end of your linearized vector, which are shown in red and yellow here, because the same 15 base pair sequences need to be added to your insert, your target gene, which can, again, be any insert. And to add these overlaps, which are homologous to the vector, you use gene-specific primers, including these 15 base pair extensions. So you just need to know the 15 bases found at either end of your linear vector and add it to your insert. So once you purify the insert and linearized vector, you simply incubate it with the infusion enzyme for 15 minutes at 50 degrees C. And what the infusion enzyme does is it fuses the insert to the vector at these 15 base pair overlapping regions. The infusion cloning reaction is then transformed into E. coli, and you get positive clones containing your recombinant construct. So it's really nice, and the great thing is you don't have to worry about the sequence of your insert because you never digest your PCR insert. You simply just add the 15 base pair overlaps, which are homologous to the overlaps on your vector. Okay, so let's take a closer look at how this works exactly. So if you imagine that this circular vector has already been linearized here, so this black sequence here is missing, you can see that the 15 bases found at either end of the linearized vector have already been added to the insert. So when you incubate the insert and vector with the infusion enzyme, what happens is the infusion enzyme actually uses its 3' exonuclease activity to create single-stranded regions at the ends of the vector and insert, which allows them to spontaneously anneal at these 15 base pair overlapping regions. Okay, and when this annealed molecule is then transformed into E. coli, any gaps are then repaired, and you get generation of your positive clones containing your recombinant construct. It's really as simple as that. So it's not ligation dependent at all, it's ligation independent, and it's not exactly recombination. That's a unique mechanism. And what are the primary advantages of infusion cloning? Firstly, cloning success, because we've put 10 years of progressive research into perfecting this product. It allows you to clone directly into any vector, and the cloning is seamless, so no extra bases added. And really the performance is excellent because we obtain over 95% cloning efficiency across a wide range of insert sizes, so anything from 20 base pair oligos to 15 kb inserts. So really you only need to pick 2 colonies for every construct that you try to clone with infusion. It's really fast and accurate, so everything can be done in 15 minutes. And it's simple and powerful, so you can clone longer inserts as well as single and multiple inserts in a single reaction. So it's really one system with multiple applications, and this graph at the bottom here shows that although the total number of colonies does drop as you clone longer fragments, so we've tried to clone an 8 kb, 12 kb or 15 kb fragment here, you can see that the percentage of positive clones doesn't change, so this remains high. So when we screen these 10 colonies for each of these fragments by colony PCR and then sequencing, you can see that either 9 out of 10 or 10 out of 10 were actually correct. Now this slide here shows the accuracy and the seamless nature of infusion cloning. So these are clones that we've generated with infusion. We've just shown the sequence data here. So as you can see, there are no errors. The cloning is also directional, and it's also seamless because there are no extra bases between vector and insert, because with infusion cloning you precisely define where you clone the insert within your vector, so you don't need to add any extra bases at all unless you want to. And this is particularly important for protein expression constructs because you don't want to add any additional amino acids to your protein, which could actually sometimes change the structure of your protein. So by using infusion cloning for your experiments, you avoid this. So the other great thing about infusion cloning is it's so versatile. So it's really ideal for several applications, including single insert cloning, cloning into large vectors such as cosmids or viral vectors, for multiple fragment cloning in a single step, for deletion insertion mutagenesis, for substitution mutagenesis, and also for adding small N or C terminal tags. I don't have time to talk about all these great applications of infusion, so I'll just concentrate on multiple fragment cloning and substitution mutagenesis today, and these will be covered in my next few slides. So the first really nice application of infusion is multiple fragment cloning. So you can really clone multiple fragments in a single reaction into your vector of choice. You don't need to do a series of sub-clonings to generate your construct. So how does this work? So the first step, as always, is to prepare your linearized vector. So you can do this either through restriction digestion or by inverse PCR if you want to be completely restriction digest free. The next step is to then amplify your various different fragments that you want to clone into this vector via PCR. And it's important to design the primers so that it either adds a 15 base pair overlap to one end of the vector or to the adjacent fragment, insert fragment, to which the insert will be joined to. So as you can see here, fragment 1 shown in red here, the forward primer has a 15 base pair overlap to one end of the linearized vector, and the reverse primer in fact has a 15 base pair overlap to the second fragment shown in blue here. But it's only the reverse primer of the second fragment which has an overlap to the third fragment shown in green here, and similarly just the reverse primer of the third fragment carries a 15 base pair overlap to the other end of the linearized vector. So we have a nice online primer design tool to help you with this, so don't worry if you're getting worried about the primer design. And alternatively, you could even synthesize your various inserts as, for example, G blocks from IDT. So in this case, you would just need to add the 20, for multiple fragment cloning, we now recommend using 20 base pair overlaps because this works better for multiple fragment cloning. So for single insert cloning, we recommend 15 base pair overlaps, but for multiple fragment cloning, 20 base pair overlaps are now recommended. And we will also have a specific protocol for this coming out soon. So anyway, as I was saying before, so if you synthesize these fragments as G blocks, you can easily join them. You just need to make sure that they have the 20 base pair overlaps required for infusion cloning. The next step is then to simply just incubate the various different inserts and your vector with the infusion enzyme. And the infusion enzyme will effectively clone these multiple inserts as a single insert into the linearized vector, because all the inserts will anneal at the 15 base pair overlapping regions to either one end of the vector or the next insert. So in this way, you don't need to worry at all whether the restriction enzymes you're using to digest the vector cut into your insert, because you don't treat your inserts with any enzymes at all. You just need to add the appropriate overlaps. In this way, you're able to clone multiple fragments in a single reaction with infusion, and you get a good cloning efficiency even for multiple inserts. So this is really nice, and don't just take our word for it. We have a nice publication for this, which you can look at. And also, this is much better than traditional ligation-based cloning, because if you wanted to do this with traditional ligation-based cloning, you would often face restriction site availability problems. You would probably need to clone in each insert sequentially and sequence in between each insert. So you would have many sub-cloning steps. Whereas with infusion, you wouldn't need to do this because you're directly cloning all three inserts into your destination vector. And because the cloning efficiency is so high, you don't need to screen many colonies either. Okay, so this is just some data that we have for this multiple insert cloning reaction. So here we try to clone the MBP Prof 12 and ACGFP fragments into linearized PUC 19 to generate a final construct of 5.2 kb. And as you can see, when we set up an infusion reaction with the three inserts and vector containing the appropriate overlaps, we obtain 89 colonies in total on the plate and only one colony on the negative control. But when we check 26 out of the 89 colonies by colony PCR and then sequencing, we obtain 100% cloning accuracy. So really 26 out of 26 were correct for a multiple fragment cloning. So you can see that the total number of colonies is, for example, less than what you would get for a single insert cloning, but still the percentage of positive clones is high. And now with our 20 base pair overlap protocol for multiple fragment cloning, you will obtain even more colonies than this, because this was done using 15 base pair overlaps in this experiment here. Another great application of infusion is in fact substitution mutagenesis. So here, if you already have a gene cloned into a specific vector and you just want to make, for example, a point substitution, you simply just choose the site to be mutated, so it's shown in yellow here. Then you just need to linearize this vector by inverse PCR. So you design your inverse PCR primers in such a way that the 5' end of each primer not only overlaps with each other, but also contains the region, the bases to be mutated, so which falls within this yellow region that you've decided that you want to mutate. So once you've generated your linearized vector this way, you simply then use infusion to recircularize the vector. So we have a nice Nature Methods app note which explains this, but it's really a great application for infusion because you don't need to buy a separate site-directed mutagenesis kit for your mutagenesis experiments. And you can do deletion and insertion mutagenesis in a similar fashion, but I don't have time to go into this in my talk today, but you can find out more on our website. Okay, so we really believe that Infusion HD Cloning Plus, which is the latest formulation of our infusion kit, is the cloning solution for really trouble-free, consistent cloning. So what comes inside the box is our proprietary Infusion HD enzyme cloning premix. We also provide a high-fidelity PCR premix clonamp Hi-Fi, which has one of the highest fidelity on the market, and it's also fast, so it has an extension time of 5 seconds per kb. We also provide an insert purification kit containing nuclear spin columns or cloning enhancer if you get a pure PCR band. And we provide our highly competent stellar cells, which are really ideal for use with infusion because you have the best cloning efficiency with our stellar cells. So by providing all these various subcomponents within our Plus kit, you will get a high out-of-the-box success rate with our system. And equally outside the box, we provide expert technical support. So if you get any if you have any problems with your cloning, you can contact our tech. And we're continually innovating our products and we are dedicated to our customer success. And in fact, our Plus system is a value packaging, because if you were to purchase each of the components separately, it would be much more expensive. Okay, so let's go back to the Clone Express and Purify workflow. So now that you've generated your target protein expression construct, the next step is to express the target protein. So as I mentioned before, Takara offers a wide range of systems, but now I just want to focus on the Brevibacillus and B. satilis systems that we offer, because they're really a bit different and a bit unique to Takara. Okay, so what's nice about the Brevibacillus system is you get the protein being secreted directly into the medium, and you often get better results than E. coli. This is because in Brevibacillus you can have disulfide bond formation. You also have no endotoxins, which is in contrast to E. coli. And another good thing about Brevibacillus, because it doesn't stay in the cell long, it's good for toxic proteins. So it's just secreted quite quickly into the medium. And in fact, with our current Brevibacillus systems, we offer 2 different vectors. So if you still prefer to express the protein intracellularly, you can do this by just choosing the vector without the secretion signal. Or if you want the protein to be secreted, just choose the vector with the secretion signal. So yeah, and also it has a low protease activity, Brevibacillus, so you often get your active protein secreted, so you can do your downstream functional assays quite easily. And we actually have 2 different Brevibacillus systems. We have our traditional system, which where you first clone into the shuttle vector, so you clone into the Brevi vector in E. coli, and then after that you transform it into your Brevibacillus cells. Or alternatively, you can use our new BIC system. So here you directly clone into the Brevibacillus vector, so you save an extra cloning step. You don't need to use a shuttle vector basically. And in fact, the BIC system, this stands for Brevibacillus in vivo cloning. So it looks a little bit similar to infusion, but it's a little bit different. So here we provide these PBIC vectors as linearized vectors, and you simply just add 15 base pair overlaps to your target gene, which are homologous to the ends of these linear vectors. So we give the overlap sequences in the user manual. But then you actually directly transform the linearized vector and insert into Brevibacillus, and here you get cloning into the vector in vivo in Brevibacillus by recombination. So it's a little bit different to infusion, but still it's a nice system. And then you just pick your colony, and then you basically grow it up and collect your protein from the medium. Now the other really nice system that we offer, which is a bit different and better than E. coli for many things, is our B. satilis protein expression system, where you can really optimize the secretion of your target protein. So similar to Brevibacillus, the protein gets secreted into the medium here. The yield is very high, and because it's a safe organism, you can really scale up your protein expression. So if you use our system, you get nice expression of your target protein. You can then easily scale up in fermenters, in medium and large scale fermenters, using B-Citillus because it's a safe organism. So what's nice about our system is actually we provide not only a B-Citillus strain, but we provide a library of different signal peptides for Brevibacillus, so namely 173 different signal peptides, so it's like a little library. And we provide, of course, the expression vector, which is this PBES vector here. And in fact, this PBES vector has this subtilisin promoter, and this drives expression of your target gene. So the first step is to clone your target gene into the multiple cloning site. So you can do this either via traditional cloning, or even better, you can use our infusion system. So once you've cloned this, here you just need to make sure your insert doesn't contain the MLU1 or ECHO 521 sites, because after cloning your target gene, you then digest out the signal peptide here, shown in green, this APRESP, using MLU1 and ECHO 521. Then you do a library scale cloning with infusion. So you basically generate a library of your specific target protein with different SP tags. And then you screen your various clones to see which SP tag gives the best secretion of your target protein. So it's a really nice system to optimize your secretion. Okay, so that leads me nicely to our, and one thing, sorry, I forgot to mention is that this vector also contains a his tag, so it allows easy purification, downstream purification of your target protein. And for this purification of his tag proteins, I now want to introduce you to our next generation Capture-M technology, which is a really fast and easy way. For example, if you think back to the B-Citillus system I mentioned, where you want to screen the different signal peptides to find the one that gives the best secretion, you could use our Capture-M technology to screen your many clones very easily, because it's very fast and easy. So what's so great about this Capture-M technology? So Capture-M technology, so this consists of a high-capacity membrane. So we have a high-capacity nickel-containing membrane for his-tagged protein purification. But we also provide kits with a high-capacity protein A membrane for native monoclonal and polyclonal antibody purification. So I'll go more into what a high-capacity membrane is in my next slide. But what's really nice about our Capture-M technology is first that it's very fast. So the mini prep protocol can be completed within 5 minutes. So as this picture at the bottom shows, it's really as easy as equilibrating the column, binding your clarified lysate, then doing a wash and elution step, and you're done. And all this just takes 5 minutes with the mini prep protocol. This becomes 15 minutes if you use our 96 well plate or maxi prep columns. But because it's a membrane-based technology, it's completely resin-free, so it's not as laborious, and it can be done at room temperature rather than at 4 degrees C. And the great thing about Capture-M is also it's compatible with many additives and lysis buffers, because your protein doesn't actually sit on the membrane that long. So for example, you can use EDTA and beta-mercaptoethanol up to a certain concentration, and I'll go through this in my later slides. And the other really nice thing is that you obtain a very highly concentrated protein, because the bed volume is small, so you can elute in a small volume, so you don't need to concentrate your protein afterwards. And we provide, as I said, mini prep, maxi prep, and 96 well formats of our kits. So it allows fast and easy purification, and our mini prep and 96 well plates are ideal for fast screening. If you have an experiment where you want to screen many protein expression clones, this would really be ideal, because you save a lot of time and money too. So this is how the plastics look like. You can see the mini prep, the maxi prep, and the 96 well plate. Okay, so now let me explain a bit about how our membrane technology is better than current resin-based purification columns. So resin-based columns, although they're high capacity due to the large internal surface area, they have a large bed volume. So this means that you get a slow diffusion of macromolecules through the resin, through the bed, and at least a long separation time. So although you get a high yield, it takes a long time. But we do have such products. We have a His 60 nickel and talon resin. So we do provide resin-based columns if that's what you prefer. But I hope today that I can convince you to try our Capture-M membrane-based column. So traditional membrane-based columns were plagued with low capacity. So even though you get a rapid flow with membrane-based columns compared to resin-based column, most traditional membranes have a low capacity. But with our Capture-M technology, we've modified these membrane pores so that they're high capacity, so they have a large internal surface area. So in this way, you still get a good yield of your protein, but you get a very fast purification or a rapid flow-induced mass transport due to the small bed volume. And there's also a low pressure drop. So the small bed volume is also nice, also ensures that you don't trap too many contaminants. Okay, and also the wash, you use 300 times more wash solution compared to bed volume, so you get a much purer protein at the end. So really, this is something quite unique in the protein purification field, this high-capacity membrane. And if you would like to test our technology, we do have samples for both the His-Tag and Protein A antibody mini prep. We have a 2-column sample, so you can contact me at the end of the webinar and we can organize a sample to be sent to you for testing. Okay, so now I just want to show you in this slide that the protocol itself is also very simple. So first I'll talk about the His-Tag mini prep, His-tagged protein purification kits. So the protocol is essentially the same for the mini prep, 96 well plate and maxi prep. It's just the volumes will be larger for the maxi prep, and also the incubation time or the spin time on the column will be slightly different. So the first step is obviously to prepare your clarified lysate. So first you start off with you can start off, for example, with a bacterial culture, or you can use mammalian cultures too, or you can in fact directly use culture media as well as your source of protein. So the first step is then to spin it down to clarify it so that you don't clog up the membrane. Sorry, the first step is to obviously lyse the cells and then clarify it so that you don't clog up the membrane. You then equilibrate in the meantime for example, your mini prep column. By adding your equilibration buffer, you do a 1-minute spin. This becomes 3 minutes if you use the 96 well plate or maxi prep, and this is similar in the downstream steps too. So then you bind your clarified lysate to the equilibrated mini prep column. So this is a really important point as well, because you don't need to incubate your sample on the membrane for any additional time. You don't need to really bind it to the membrane because the binding is immediate. So you add it to your column and you spin immediately for 1 minute or 3 minutes. And then you transfer the column onto a new tube and you wash. Again, add the wash buffer and again spin for another 1 to 3 minutes depending on whether you're using the mini prep or the maxi prep. And then finally you elute, you transfer it into a new tube and elute in a small volume. And again, you do this via a 1 to 3 minute spin step. So you can see that the whole protocol can be completed in 5 minutes for the mini prep or in under 15 minutes for the maxi prep and 96 well plate versions. But then after you've purified the proteins, if you then run it on a gel and Comasi stain it, you can see here in the stain that you get really quite nice pure protein, and it all comes out in the first elution, so nothing stays on the membrane. But the important point to remember is the binding step, that the binding is immediate and you don't need to waste any extra time binding your target protein to the membrane, because this can be quite a time-consuming step with the resin-based column, where you'll have different incubation times depending on the nature of the target protein itself. And here, this table on the right importantly shows you the yield expected. So from the mini prep, you can expect to get a yield of 100 micrograms per column, and this is the same for each well of the 96 well plate. But this yield increases to 2.5 mg per column with the maxi prep. Okay, so in this slide, this slide nicely illustrates that our Capturum Histag kits are compatible with additives. So you can use additives such as EDTA or beta-mercaptoethanol or TCEP or DTT within your lysis buffer. This is possible because with the Capturum membrane, the protein doesn't sit on the membrane for too long. So you get away with using these additives in the lysis buffer and also in your wash buffers if needed. So here you can see that the Capturum Histag membrane can tolerate up to 10 millimolar EDTA, which is a common additive that's used in protein purification studies. And normally researchers would have to do kind of a buffer exchange to get rid of this, but with Capturum you can just leave it in. And similarly with beta-mercaptoethanol, you can use up to 30 millimolar of beta-mercaptoethanol. So you can see, again, you get a nice clean iluate in iluate 1. And the same is true for up to 10 millimolar of TCEP and also 10 millimolar of DTT. And we have tested many other additives, including non-ionic and anionic detergents. And this is shown in the table on the right here. So, Capturum, due to the structure of the membrane itself, this high-capacity nickel membrane, it's particularly useful for the purifications of hard-to-purify proteins, such as G-protein coupled receptors. So, G-protein coupled receptors, researchers who work on these proteins usually face the three main challenges, namely low expression levels, high hydrophobicity, and also low stability in detergents. But these challenges are not just specific to GPCRs, but they can also apply to other difficult to purify proteins. But how can our Histag purification kit help with these issues that researchers face with GPCRs? Okay, let's tackle each one by one. So for example, if you have low expression levels of your protein, traditionally most researchers would get around this by expressing your protein in a high volume to get a sufficient quantity. But then this takes a long time, you have to use a lot of resin, and then also you might end up with a very dilute iluate, okay, in the end. So you may need to also concentrate your protein afterwards. But if you instead use our Capturum Histag Maxi Prep, you can in fact pass a high volume of lysate. So with the Maxi Prep column, you can do 2 loads of 150 mils of bacterial culture, or medium or even mammalian culture, and in fact, you can elute this. You can pass a high volume, but you purify a concentrated protein in a small iluate. So with the Maxi Prep, you can elute in just 0.5 ml of elution buffer. So this is a nice way to get around this issue of low expression level. Now, what about the problem of high hydrophobicity? So GPCRs are not very soluble. So normally researchers get around this or try to resolve this problem by using detergents or other additives to make the protein soluble. Now this is a bit complicated when you're using resin-based columns, because you would add the additive to your lysis buffer, but then you would need to do a buffer exchange before the actual resin purification, because the additives can destroy the resin. And so this can be problematic, whereas with Whereas with our Capturum high-capacity membrane, you can use the additives directly in the lysis buffer and just leave it, because the protein doesn't sit on the membrane for very long. Okay, so there's no need for buffer exchange. There's no need to use a large amount of resin, and in the end you get a concentrated protein because you're able to elute in a small volume. And the other challenge of low stability in detergent. So for GPCRs, because you would use detergent to overcome the high hydrophobicity, what sometimes happens is that the protein stability itself is affected, because most proteins are sensitive and have a low stability in detergents. But because Capturum technology is fast and the protein doesn't sit on the membrane for very long, the protein time in the detergent solution is actually minimized to minutes, and you get an improved reconstitution of the protein after purification. And in fact, you get improved protein functionality compared to resin-based purification systems. So in this way, you can use our Capturum his-tagged kits to purify GPCRs, which are difficult. So we actually did do this experiment in-house where we purified a GPCR with our Capturum his-tag mini prep. Okay, so what did we do? We actually expressed this ADRB2 receptor, so as a his-tagged with the 6 his-tag. So this is the adreno beta 2 receptor, which is a GPCR, in 293T cells. We then prepared a lysate from the 293T cells and clarified it and applied it to our mini prep column and followed the protocol as normal. Equilibration for 1 minute, with then binding, washing, and eluting in 1 minute. And as you can see, after the Kumasi stain gel, after the SDS page gel was blotted onto a membrane and the membrane was stained with Ponceau Rouge, you can see that this ADRB2 receptor is detected in all the fractions, so the lysate, the flow through, the washer, and the eluate. But when you then do a specific Western blot with an ADRB2 antibody, you detect it. You can see that it's concentrated in the eluate. So we were able to purify a GPCR in 5 minutes, once you have the clarified lysate ready, which is quite a feat. And so you can really use our Capturum products for difficult to purify proteins. If you're having, for example, issues using resin-based purification, why not try our Capturum mini prep sample? Similarly, we also have Capturum protein A purification kits for antibody purification. So here you actually purify the native monoclonal or polyclonal antibody. So it's the same principle as the his-tag purification kit. The only difference is this time the high-capacity membrane contains protein A rather than nickel. So protein A is able to bind the FC region of IgG or antibodies. Okay, and that's how you're able to purify. And again, it's a high-capacity membrane, which allows a good yield but fast purification due to the low bed volume. So I won't go into that in too much detail again because I already explained that before. Now, in terms of the protocol, it's again simple and quite similar to the his-tagged mini prep, maxi prep 96-well protocol. The only difference is obviously you're adding your diluted antibody sample onto the equilibrated column here. So this can be you can use serum directly. You can dilute the serum and use it, or you can use asite fluid. Also, or cell culture natant, supernatant directly, so or other sources. So it's really quite flexible. So again, after loading it onto the equilibrated column, again you get immediate binding of the antibody to the membrane. So you don't need to optimize binding of the antibody to the membrane, which can be an issue with other more traditional resin-based antibody purification systems, which usually also contain a protein A-based resin. Then again, you do a wash step and an elution step. So when we did this experiment with human serum, you can see that after elution we get a nice clean antibody purification. So this is the heavy IgG heavy chain, and this is the light chain here. And in terms of yield, the mini prep yields 300 micrograms of antibody per column. 96 well plate, you get 300 micrograms of antibody per well, and the maxi prep gives you up to 2.5 mg per column. But of course remember that the antibody yield will also vary upon the species of the serum. And that brings me to the next slide. So here we show that our capture protein A mini prep binds to antibodies from various species. So here we directly used animal sera on the capture mini spin columns. So you can see that we were able to successfully purify antibodies from different sera, including sheep, goat, rat, mouse, human, rabbit, and horse. And you can see the amount varies according to the species. And another really nice application of our capture protein A mini prep, but equally you could also do this in high throughput in the 96 well plate, because each well of a 96 well plate is equivalent to a mini spin column. Another really great application is that you can do fast and efficient immunoprecipitation directly on our protein A mini prep column. So how does this work? So for example, if you're studying a specific protein and you want to purify it with a specific antibody that it binds to it. For example, here we did this with the PP2A beta subunit. So you add your specific antibody to lysate from cells expressing your specific target protein, in our case the PP2A beta subunit. You mix it. You already mix it for 10 minutes in a separate tube, and you do end-to-end rotation on a wheel. So normally an IP, it can take many hours to even overnight incubation, but with our mini prep you simply incubate the antibody and the lysate for 10 minutes. So after this 10-minute end-to-end rotation and this can also be performed at room temperature, you simply load this mix onto an equilibrated mini prep column. Again, this is then bound, then you do a wash and elution step. You just follow the normal mini prep protocol, and when you analyze the iluate in a Western blot, you can see that we were able to purify this PPTA B subunit. So the important point to remember here is what the membrane is capturing is the antibody for the PP2AB. It's binding to the FC region of the antibody. Okay, and because the protein itself is bound to the antibody, you're co-purifying the protein. You're purifying the immunoprecipitated protein. And this is really nice because normally this would take many hours to even days, but with our capture mini prep protocol, you can do this within I guess a few hours, or yeah, fairly within an hour once you have your lysate ready. And it's a similar process if you also want to do fast and efficient co-immunoprecipitation. So co-immunoprecipitation is usually used to detect in vivo protein-protein interactions. So if you want to find out if protein A binds to protein B in vivo. So what you normally do is you use the antibody for protein A, or you can use the antibody for protein B to do an initial immunoprecipitation of a cell lysate expressing both protein A and B. And then you can just follow the immunoprecipitation protocol that I just mentioned here before. The only thing to remember is, so you use the antibody and incubate it with the lysate for 10 minutes with end-to-end rotation at room temperature. Then you load it onto the equilibrated column. So when you bind it to the column captures, again, the Fc region of the antibody, but it will also capture the protein that the antibody is bound to, right, which let's say is protein A here, but you will also co-purify protein B if it's strongly interacting with protein A. So you're doing a co-immunoprecipitation. So we did exactly this experiment for P53 and SV40, which are known strong interactors. So here, in fact, we expressed SV40 and P53 both in 293 T cells, and we used 293 T cells or HT1080 cells in this experiment. So 293 T cells are known to express endogenous P53 and SV40, whereas HT1080 cells don't express either of these proteins. So then we either used the SV40 antibody for the pull down or the P53 antibody for the initial immunoprecipitation. But you can see that when we blot for the other protein, that both proteins were pulled down. So both the SV40 and the P53, which is what you would expect if these proteins are really interacting in the cell in vivo. And similar story, if you just immunoprecipitate with the P53 antibody, you also pull down SV40. And this is what's shown in this Western blot here. And the HT1080 cells give the expected results because we know they don't express either P53 or SV40, so you don't get any bands here. And you can read more about this by clicking on the link within this slide, which is a nice technical note explaining how we did this. So as I was saying before, our Captura Mini Prep and 96-well plates are ideal tools for screening many proteins because it's very fast, it's easy, and it's the 96-well plate is suitable for high throughput applications. So if you want to do many samples in high throughput, you can use that. But remember, the amount of protein that you get isn't huge compared to resin. So if you want a lot of protein, it's not suitable. So you may still need to scale up if you want a large amount of protein. So I would suggest that you consider using our Captura Mini and 96-well plate for screening purposes. And then if you really want to do a large-scale purification, you can then scale up to our His60 nickel resin or our Talon resin if you want purer protein for crystallography studies. So I'll just briefly mention these two products that we offer. So these are classical resin-based purification systems. So we offer our His60 nickel resin, which gives a very high yield. So 60 has a binding capacity of 60 mg per mil. Or you could consider, so if you use our Captura Histag Mini Preps, you can easily scale up to the His60 nickel resin because it's the same chemistry. But however, purity is a little bit compromised with the His60 nickel. Because it's balanced for yield and purity. So if purity is more important than yield to you, then you could instead consider using our cobalt-based Talon resin. So here, this has a lower binding capacity of 20 mg per mil, but it's really, you'll get very, very pure protein that you can then directly use in your crystallography studies. And we have a choice of formats for the different purification scales. So we have bulk resins, magnetic beads, FPLC cartridges, which can be used in ACTA machines, as well as gravity and spin columns. So yeah, this just picture shows the different formats that we offer. So for example, most people buy our bulk resin and pack it into their own columns. So you could do this, or you could buy our readily packed cartridges. So if you have an ACTA machine, you can buy them different size cartridges for this, or you can use our gravity columns. Okay, so I just wanted to summarize by going back to the clone, express, and purify workflow. So I hope today that I've shown you some of the next generation tools that we offer for protein expression and purification, really for the entire workflow. So everything from cloning your gene with infusion to expressing your target protein, for example, with Brevibacillus or Bacillus, and then purifying the target protein. So doing an initial screening with our Capturum technology, and then easily scaling up to our bulk resins, His-60 nickel or even Talon. So if you have any more questions, please just type them into the chat box. I'll go through the questions that we already received. But you can also visit our website. So we have a cloning resource portal where you can find out more about infusion and the different applications that it's been used in. Or you can visit our Protein Learning Center to find out more about the different protein expression and purification products that we offer. Yeah, I also forgot to mention that we have samples also for the Talon and His-60 nickel resin. So if you're interested in testing this, you can also get back to me. So I'll just go through the questions. Oh, sorry. And finally, you can also email us at tech@takara-clontech.eu if you have any additional technical questions, and we'll be happy to get back to you. So let me just go through the questions now. Okay, so one question was, are there any special yeast expression systems? That's a good question. We don't actually offer any yeast protein expression systems. We offer our Matchmaker Gold yeast-2 hybrid screening system, but these are really just for yeast-2 hybrid screens where you're detecting protein-protein interactions. And we also offer yeast-1 hybrid screening products, but yeah, sorry, unfortunately we don't have a specific yeast expression system. Okay, another question. Can you comment on the difference to Gibson assembly? Yes, so you're talking about infusion compared to Gibson. So Gibson, it is a similar to seamless cloning technology, but the only difference is that it also uses a 5' exonuclease activity, and it also consists of 3 different enzymatic activities. Okay, so the first one is the T4 polymerase, which chews back, and then they have a polymerase which fills in the gaps, and then they have an in vitro ligation step before it's transformed into E. coli. Whereas with infusion, you have a 3' exonuclease activity, and there's no filling in with the PCR polymerase in vitro. Rather, the gaps get repaired when the infusion cloning reaction is transformed into E. coli. So you have in vivo gap repair happening with infusion. The consequence of this is you get less background with infusion compared to Gibson. So with Gibson, we have done a side-by-side comparison, and you can find out more information on our cloning resource portal. There is a special flyer for this, or I can also separately send this to you by email. And we tested Gibson compared to Gibson for both single insert and multiple insert cloning. And basically, although for both you obtain more colonies with Gibson, the percentage of positive clones is lower than with infusion. And we think this is because of the in vitro ligation step that you have with Gibson assembly, which you don't have with infusion. So if you're referring to the NEB kit, but if you're using the homebrew Gibson assembly method, then again, it's not a QC'd kit. So that's the advantage infusion has. So yeah, you need to screen more colonies with Gibson, so I would recommend infusion. And another question, how does the elution buffer look like in a classical His-tagged based affinity chromatography that one would use imidazole containing buffer? Is this the case with your system? If yes, what do you recommend for final buffer exchange? Okay, are you talking about the elution buffer within Capture-M? Or yes. Okay, yes, it is quite similar to the one that you would use for with our His-60 nickel resin. So customers can use their own, but we have specific recommendations which you can check in our user manual. So yeah, the whole idea is that you can easily scale up to the His-60 nickel resin because it's a similar chemistry. And another question, which is the binding capacity of his tagged mini prep and maxi prep columns? So it's equivalent to 75 mg per ml. So it has much higher binding capacity if you compare resins. I can send you a poster about this which explains this better. Okay. I think that those are all the questions now, for now. Okay, so we'll be sending you all a PDF copy of the presentation from today, as well as a link to the recorded version of the webinar. So in the meantime, if you have any further questions, you can just email our tech support address. Okay, thanks again for joining today's webinar, and I wish you a good day ahead. Thanks. Bye.